Good morning, everybody. Good morning to all. I am Dr. Prashant Kare. It gives me immense pleasure to welcome you all for the 2 days webinar on advances in basic plant researches in relation to climate change. This is organized by Department of Agricultural Biochemistry and Center for Advanced Agricultural Science and Technology for Climate Smart Agriculture and Water Management, Mahatma Phule Krishi Vidyapit, Rahuri. Before this, I have some announcement. Participants who have not joined by their name are requested to rename themselves because it is important for recording attendance during session and also to recognize the registered participants. Secondly, if you have any question or queries related to topic during the session, uh, uh, you can write your questions in chat box. Towards the end of the session, we will answer these questions with the help of experts. Please resist yourself from posting good morning messages in chat box. Another one, we shall post short assignment on WhatsApp groups. Participants are requested to submit the assignments on time uh, as per the guidelines issued. During these 2 days webinar series, we'll have 4 sessions, that is 2 sessions each day. I would like to welcome all the participants on Zoom and YouTube platform from different parts of the world. In the short inaugural function of this webinar, I welcome to the experts of the session, university experts, organizing team members, CAST team members, research associates. Now, I would like to request Dr. Pawan Kulwal sir, Joint Organizing Secretary and Professor of Agricultural Botany, to welcome and preamble of the webinar series.
Thank you, Prashant. I think before that, maybe Dr. M.G. Shinde would like to tell about the caste, and then I will tell about the sir.
Yeah, okay. Thank you, Dr. Kulwal. Dear respected dignitaries and all dear participants, heartily welcome for this webinar series, 2 days webinar series. For the information of dignitaries and the participants, this webinar series is being organized by Department of Biochemistry and Center for Advanced Agricultural Science and Technology for Climate Smart Agriculture and Water Management. This is a World Bank aided project through Indian Council of Agricultural Research under National Agricultural Higher Education Project. The major objectives of this project, they are to build the capacity of the students of Mahatma Phule Krishi Vidyapit, Rahuri, and also the faculties, scientists, and also the farmers in relation to climate smart agriculture and water management. So to meet these objectives, we have been conducting series of trainings, workshop, webinars before the lockdown, and after the lockdown, we started on the we switch over on the online mode. So just we completed 50 trainings, and this is the 51st event of this series of events. Then we also have introduced a few courses for MSc, MTech and PhD programs. The courses are related to, again, climate smart agriculture and water management. We have introduced 7 new courses, and those courses will be taught from this current academic year. We are also starting 1-year postgraduate diploma in climate smart agriculture and water management. The diploma is module based, and it is more practical oriented, and the students who registered for this postgraduate diploma, they are expected to be experts, more experts in practical skills than theoretical skills, and they will be observed in different organizations who are working on the climate smart agriculture. We are also starting, or already we have started, postgraduate postdoctorate program, and we are receiving queries for this postdoctorate program. That program is already being started. Then we are also developing innovative technologies based on remote sensing GIS, which can offer good solutions to the farmers on different aspects of climate smart agriculture and water management. Of these technologies, and these villages, they are being developed as digital villages. We are installing automatic weather stations and other instruments like real-time soil moisture sensing and all these kind of things, and we are extending these technologies which are being developed at the campus. So with this brief introduction about the cost, I request Dr. Kai to proceed.
Thank you, sir. Dr. Pawan Kulwal, sir, please.
Okay, thank you very much. Good morning to all. Let me first welcome our expert speaker, Dr. Sheshshayee, Dr. Chinnuswamy, also from IR, and Dr. Tahuja. I could see them all online. So good morning to you all, sir. So regarding this webinar, just want to tell briefly that as part of the climate smart agriculture, the theme of our project, CAST project, we have started a webinar series from July 22nd, which was the 198th birth anniversary of Gregor Mendel, the father of modern genetics, with the lead lecture from Professor P.K. Gupta. And in that series, we are conducting this another webinar series on advances in basic plant research in relation to climate change. So as part of this program, we received a huge response from participants from across the world. The total number of registrations we had is 1,965, and it representing 10 countries, including India. So participants are from Germany, Ghana, Nigeria, Cameroon, Algeria, Nepal, USA, Australia, and Israel. So in terms of gender distribution, out of these 1,965 participants, we had 703 applications from female candidates, as well as 1,093 applications or registrations from the male candidates. And it comprised of more than 500 students and equal number of faculty members from across different parts of the country. Almost all the states we have representations, and many will be joining through YouTube platform. So with this background, now I request Dr. R.M. Naik, who is the organizing secretary of this event, to tell brief about the background of this webinar and introduce the guest. Over to you, sir.
Thank you, Dr. Kulwal. Good morning, one and all. And I take this opportunity to welcome our expert speaker, the participants, dear participants. Today's webinar is proposed because, as we know, that the development in the field of basic science, plant science research has significantly contributed in understanding the metabolic network and the key targets for improving the crop plant. And the dependence of cell organelle, the crosstalk, and the interdependence of the cell organelle has been worked out at the cellular level. The basic research in plant biology is more important today than ever, considering the escalating pressure of food, fuel, fiber, and the plant products, the climate change, malnutrition, and human health, and many important questions, many important you can say questions have been partly or fully answered during the last decade by the omics platform, like how do plants integrate multiple environmental signals and response, what is the role of microRNA, are endogenous plant adaptation mechanisms enough to keep up with the pace of the environment change? To what extent do epigenetic changes affect heritable characteristics of plants? And like some of the questions like improvement of T3 field through incorporation of T3, T3, T4 or SAM mechanism, and what is the biochemical or molecular basis of the drug? So some of these questions have been partly answered, and a success has been achieved, a breakthrough has been achieved through a systematic effort through different omics platforms, and that is why there is immense need to concentrate on the basic research. The development of transgenic crops by transfer of alien genes has contributed significantly. However, some of the controversial results need consideration of the regulatory processes such as post-translational regulation, substrate limitation or product inhibition, and many question marks are still there. But we through this platform, that is advances in basic plant research in relation to climate change, through this platform, we have invited the experts who have immensely contributed in the field of basic plant research, right from the development of the mutant and deciphering the water deficiency or nutrient deficiency to the genome editing, the application of genome editing in crop improvement. So that was the purpose of hosting this particular, selecting this particular topic. And I am extremely grateful to our Honorable Vice Chancellor, the team and directors, and the CAST principal investigator and co-PI of the CAST and all the CAST team for approving this topic of the webinar. I'm here to introduce uh, today's expert, Dr. M.S. Sheshshayee, who is a professor at Department of Crop Physiology, University of Agricultural Sciences, Bangalore, since 2010. Dr. Sheshshayee has many honors and awards. Few of them are he is recipient of the Biostat postdoctoral fellowship and visited Canberra University, Australia. He, he has been awarded Outstanding Research Performance Certificate by DSP Young Scientist. Dr. Sheshshayee is a chief scientific investigator IAEA program on water use efficiency. He is member of the RCGM. Dr. Sheshshayee is the Best Teacher Award. He is recipient of the Best Teacher Award by Indian National Science Academy. He is the elected fellow of National Academy of Sciences. Dr. Sheshshayee has contributed professional experience is that application of stable isotopes in understanding plant physiological processes, which he got this while doing his postdoctoral research, development of DNA markers at CCMD. Then Dr. Sheshshayee has more than 200 research publications in good mass-rated journals. He developed state-of-art mini light-emitter-based genomics platform for precise imposition and monitoring of soil moisture status. Developed a trait-in-progress genotype of rice with reduced water requirement for a given yield potential. Dr. Sheshshayee has discovered crucial regions governing variability in water use efficiency and fruit trait and other drought adaptive traits. He has characterized the rice mutants for functional genomics, and Dr. Sheshshayee has operated more than 20 externally funded projects from DBT, GST, and this variety has been demonstrated to save 50% irrigation water and yields more than 80% of the irrigated yield potential. So such an expert is with us, and I welcome Dr. Sheshshayee, and I request Dr. Sheshshayee to continue with his presentation. Thank you.
Thank you, sir. Dr. Sheshshayee.
Yes.
Sir.
Yeah, can I go ahead?
Yes, sir.
Yes, sir. You can go ahead, please.
Thank you, sir. Thank you so much, Dr. Rajiv Naik, and for an elaborate introduction. Um, but what we have been talking about is when we look at the kind of situation that is happening, the climate is very much changing. And then we have a lot of differences in terms of both excess of everything or big shortage of resources, and with a lot of people increasing in the country, with the human population, I think there's a very important role that the agriculture scientists have to play. And I'm very happy to associate with this program of Mahatma Phule Krishi Vishwavidyalaya, Vidyapit. And I have actually visited once sometime back a few years ago, and it was an interesting discussion that we all had with the colleagues and faculty in the university. I'm so happy to this. Can I share my screen slides?
Yes, sir. Yes.
Is that visible, Prashant?
Yes, sir.
That's very nice. Thank you
Just
so much.
full screen, sir.
Yeah, I am doing that. Is that full screen now?
Yes.
You can see this. Thank you so much. And thank you for inviting me to this interesting thing. And I'm very happy also today morning, two young scientists have been awarded the Nobel Prize for genome editing. And I'm very happy that one of my very close friends, a collaborator and a co-physiologist, Dr. Viswanathan, is also here. He has also been working and pioneering the genome editing and using the CRISPR-Cas9 for drought and other things. And I'm so happy about that. In the note of that, we will go ahead with this. So I had suggested that I will talk about how to improve productivity of our croplands under the climate changing situations. Especially, I just wanted to now put this into three actual parts. We will, I mean, this is something that you're talking about is a basic science and how it can be translated. So actually, if plant physiology has ever been considered as something basic and has less relevance in agriculture field, agriculture research, but I completely beg to differ from that statement in the sense basic science has a lot of things to play, a lot of role to play in crop improvement and in applied sciences as well. So I would like to now tell you very briefly about what we have been doing on this area of how to improve the productivity in a changing climatic situations. This is something which we are aware of, that the Indian population is going to be the highest in the world, and we are going to overtake China, at least in number of people. I do not know whether we are capable of doing that in any other fields. But if you look at the productivity that we have on the right side of this graph, you can see that the yield levels are plateauing. That is one thing that we can think of. But what is disturbing to me, at least, is if you look at this graph, the kind of the blue line that you have is for United States. In California and other places, they have, whether it is rice, wheat, or maize, and all the cereals, you have a very high productivity of up to 10 tons per hectare. Whereas Indian line that you can see, it is always at the bottom. I mean, this helps me to write a project proposal to the government of India and tell that, look, we are very poor and therefore we need to improve it. But the question is, how is it that we can reach from, let's say, 2 tons to 10 tons? So there's a 5-time increase in it. And the other very important challenge that we have in front of us is how are we to feed the, like 1.5 billion people that will be in 2050. I'm sure most of the participants who are young people will still be there during that time, and in 2050, how are we going to now feed these people? Now, the other question that we have is, if you look at the Indian production, the food grain production, in terms of million tons, you can see in the last 2 decades there has not been a substantial increase at all. And though agriculture is considered as an important industry and it contributes about 60 to 75% of the people are engaged in agriculture, their contribution to GDP is very, very less. It's about 16%. That's a challenge. And unfortunately, though there is an increase in the production, the contribution to the GDP is declining. This is something that bothers us. Therefore, if you look at what is the reason for this kind of a plateauing of the yield or the kind of yield that we realize, it's not the potential of the crops. If you look at this country's scenario here, 54% of our country's regions geographically face extremely less water or extreme situations in terms of water limitation. And this seems to be the major problem that concerns us. And of course, with the increase in the temperature, there's going to be a lot of challenges that we're going to have. I mean, when I was in college, when we were students of BSc agriculture, they kept on telling in the 2010 we are going to die because there's not going to be enough food and all that. But then thanks to the breeding activity that happens in the country, there's a tremendous amount of improvement in the production. The same thing is being said now. The increment in food production should increase to something like 320 million tons. And otherwise, in 2050, we're going to suffer. But my optimism says that we will reach there. That's not a problem at all. But we should have a very strict, what you call as a roadmap for going ahead with this. So now the goal, therefore, is to sort of improve the resource capture and use efficiency. So that's exactly what I mean. So when we are trying to increase the yield, we are looking at two things. One is the resources, of course, are becoming scarce under the conditions of climate change. And when we look at that, there are two questions that we have. In the green revolution time, when there was a big improvement in the productivity that happened, mainly because of resource capture. There was resource made available, whether it was because of the dwarfing, the semi-dwarf plants where light distribution inside the canopy was better, or the irrigation projects provided the water availability, or there was some fertilization that improved the availability of nutrients to the crop. Besides genetic crop improvement, there was a tremendous improvement in resource provision, and that I was associating with the genotype that were capturing these resources. But in the present situation, when the things are going to be where the resources are going to be limiting, resource use efficiency perhaps may become very, very important. Therefore, we have two questions on hand. What do we mean by yield potential and what do we mean by the potential yields? Potential yields are those when we have no problem for resources. This is the potential that you have. And let's say 10 tons per hectare is the potential for a rice variety. But we realize about 5 times less than that because of so many stresses. That is, we have resource limiting situations in this. Now, how is it that we can either increase one of them or increase both these kind of things, potential yields as well as the yield potential? Therefore, I believe that the success depends on understanding what causes the yield potentials to increase or the potential in yields to be maximized. And therefore, we need to look at specific traits which are physiological or morphological or any of those kind of traits that we look at and capture genetic variability in them. So therefore, there are two challenges. One is, what are those traits and how are we going to capture the genetic variability for the activities in terms of breeding? So before we go into that, we need to understand what really happens when water, one of the major things, as I said, though there are, we can say that the resources are limiting, one of the major things that is limiting is water all over the country and perhaps all over the world. It could be the same situation everywhere. So when we look at what happens to the water, whenever the plants experience water stress, there is no doubt that the yield decreases. But if we look at what happens to each of the different stages or different phenological phases of the crop plant, then we can look at the yield limitation in the vegetative phase, or which we call the GS1 phase, is happening because of the reduction in the leaf area or leaf expansion, elongation, development of the leaf, and so on and so forth. There are a lot of signs that can be done. There are a lot of genes that have been discovered already, QTLs have been discovered already for the leaf size, leaf development, and so on. But if the stress occurs during the flowering to the pollination and that period of time, and the yield loss is perhaps the maximum when the stress occurs at the GS2 stage, which is definitely possible. And one of the major reasons for that, for the reduction in the yield during the GS2 is the increase in the spikelet sterility. So is the spikelets, here if I look at the percent reduction in spikelet fertility and the percent reduction in yield, there's a very strong relationship between them. And if the stress occurs during the grain filling period, which is also possible, which can happen very, very frequently in real life situations, what happens is the leaf area is already maximized, and then the pollination is over and the grains are filling, but there is not sufficient amounts of carbohydrates allowed are going available for the grain filling. Therefore, canopy photosynthesis becomes an important issue during that period. And you have a big challenge here. In one plant, during the 3 phases of its phenology, you have tremendously different ways in which the plants get affected in terms of the reduction in the yield because of stress. Now, the question is, how are we to now understand these things in terms of genetic variability for each of these things? How is it that we can create a stress? How is it that we can monitor the influence or effect of stress on the crop plant has always been a challenge. So the question that always occurs is, what should be the breeding strategy? Now, one thing that people have done in the past is to look for higher absolute yield under stress. I mean, there is no doubt that is the right way to do. It does not matter to the breeder whether the yield that we get at the end of the season is because of better leaf growth or better spikelet fertility or better canopy photosynthesis. He just doesn't bother, and then he just picks up the one that has higher absolute yields under the field conditions. Though the progress that we made in that is rapid, there's no doubt about it, but recent information suggests that there is a low variability among the already high yielding varieties. There is no much of an opportunity to now hybridize them and pick up the best ones. And then there's a very high genotype into environment interactions for yield per se. And because of the high genotype into yield, in genotype and environment interactions, the stability of the traits where the yield is per yield selection criterion is not very high. On the other hand, if you looked at a mechanism where we are trying to increase the drought adaptive mechanisms by specific traits, though the progress is slow, but because we have a very low G by E for a specific trait, I'm going to show you what I mean by that. And then these traits are highly stable. Therefore, we believe a trait-based mechanism or trait-based breeding, or what we call as analytical breeding, or these days they are also calling that as physiological breeding, is a more preferable strategy for breeding. That said, we need to look at two things. One is we have to now establish that there is genetic variability, and if there is no genetic variability, we have to be able to create the variability, and then we have to capture the genetic variability by phenotyping, and then we have to now go ahead with the breeding. But it always been a challenge where people have criticized the physiologists in terms of saying that the physiological traits that we mentioned, like for example, if you look at canopy photosynthesis, how do you measure in hundreds of thousands of plants? That's going to be the challenge. Therefore, we can have the modern ways of doing things doing the breeding, in the sense, use the DNA markers, use the molecular breeding strategies, and that can be done, which I will share with you some information that we have done on this. Now, what we have is, when we look at the traits and we dissect them, there's one thing which is very, very clear. When we have moisture stress in the soil, one of the traits that is important is a mechanism by which the plants can access water, and which is by developing better root system and better root architecture. And this was a very classical paper published by Uga and others, Henry Nguyen, and I think one of our colleagues, Jagdish Rane, was also a part of this, who is now the head of the National Institute of Abiotic Stress Management in India. And this particular paper explains very clearly how a near isogenic line in the background of IR64 is capable of doing better under stress by developing better root system. And then we had two strategies of doing similar activities in our experiments that we did in our lab. One was to use a germplasm, which I'll tell you a little bit later. The other thing that we used was a big consortium of activity that we had done, that we had undertaken, which is an ongoing program even now. In the Division of Genetics and Plant Physiology at IARA, Viswanathan is also here in the audience right now. Nagina 22, a upland variety, which is a known drought-tolerant variety, was mutated by EMS-induced mutation. Ethyl methane sulfonate is a chemical which can induce a single nucleotide change. And then we developed a very large number of mutant lines, which was screened for various traits by several investigators. And what we did was to look for drought adaptive traits, and we looked for the root traits initially, and then we have a lot of other things that we've done later. But for this purpose, I just want to tell you the root story. So N22 has a root of this sort, but then there were a few mutants. There were a few mutants that had much higher roots than the Nagina 22, and these mutants were selected, and we did further experiments on this thing. So what we did was we compared the low and the high root mutants, and we can see that the high root mutant definitely had much higher photosynthesis, much higher stomatal conductance, and much higher mechanisms of surviving under water limited conditions. In when we looked at this, the high root mutants and compared to low root mutants, and we developed a mute map population, or what we call as by crossing the high root mutant with the wild type, and then we picked up the ones that had the similar segregation of the root traits, and we identified what could be the gene that is responsible for the improvement in the root traits. And the reason why I am telling that story is there is a particular transcription factor, which is a homeodomain transcription factor, which characteristically came out as a gene which is responsible for growing better root system. And that particular gene, which is called HOX10, has also been shown in human beings for the neuronal developments and all those things. There's a huge family of these transcription factors, and they have a very important role in this. Now, the reason why I'm telling that story is the basic science that leads to these discoveries can now become the mechanism by which science can be applied for crop improvement. Now, what we have done now is to use this this mutant as a parental line and cross it with low root and high yielding varieties, and we show that there is an increase in the yield, or this increase in the productivity of such plants when water is limiting. And then we did another exercise to now understand what other traits are necessary in it. To cut the long story short, there are a lot of these kind of things. We classified the drought adaptive traits into three mechanisms. One we call as the three kinds of things that we look at. Then we have the physiological traits, the morphological traits, and the root is a morphological trait. Leaf area is a morphological trait. But what happens inside them in terms of photosynthesis or respiration or transpiration, we look at them as physiological traits. There could be many, many of them. And I'm not going into the details of these things, but there is one more set of traits which we call as the cellular level tolerance traits or acquired tolerance traits. The morphological and the physiological traits are constitutive; they are present all the time. But then when there are some of these traits which only express when the stress is imposed, and when the stress is progressing, when it's mild, certain gene expression increases and improves, and that provides the kind of protection to the plant that the plant requires when the stress becomes a little more severe. And how is it that we can capture them, and how is it that we can improve them in the breeding activities is a challenge. So in other words, when we have a large number of these many traits, if you simplify all of them, there are only three things that come to mind. One is, if you have stress, water stress, especially drought stress, you go ahead with the water mining, and then once you access this water, you access them, and then you use that water efficiently, and whatever it is possible that we can have a better conservation of this water. The water conservation is another. It's an important trait that should look for when you have stress situations. So these traits are relevant. There's no doubt about it. Only when the acquired tolerance traits are better. Therefore, what we did was to now develop a strategy to increase the root traits. And initially, that we did when we got those mutants or the germplasm, the increased root traits can do one thing disastrous. What is that? That is, they can exhaust the water more quickly from the soil, and that's a problem. And therefore, we need to increase the water use efficiency. It's not just possible that the plants are capable of absorbing water, but they have to use it efficiently. And their efficiency of use and their excess of water from the soil is possible only when the intrinsic cellular level tolerance or the acquired tolerance levels are high. Now, therefore, we have a mechanism where to now look for all these variabilities. We understand how to phenotype them. And the initial thing that we did was to use the stable isotopes as a strategy to capture genetic variability in the traits that we're talking about, both root traits as well as water use efficiency. And this is something very simple. And when we have the stable isotopes, these are not radioactive, they're stable. The isotopes are because they have extra neutrons in their atom, in their nucleus, and that makes them to be an isotope, but then they don't disintegrate, they're stable forever. And you have the lighter isotope, like for example, hydrogen is a lighter isotope of deuterium, which has one extra neutron in the atom, but it remains, and the hydrogen is elementally exactly the same thing, but chemically they have differences. I'll tell you how. And we have 3 or 4 very, very important elements for which the isotopes are useful in biological sciences. The carbon-12 is the predominantly available carbon isotope in the atmosphere or in the ecosystem. But then you have a small portion of about 1% of the carbon that you have is a heavy isotope of carbon, which is called a 13 isotope, 13 carbon. Similarly, you have nitrogen 14 and nitrogen 15, and oxygen you have 16, 17 and 18. 17 has very, very less quantity, so you have the 18. And these elements or isotopic compositions are generally used in understanding the plant physiological processes. I mean, I mean, this can go on. This is a very, very specialized topic, but then to make it very simple, what I want to say is, when you have the kind of distribution of these elemental compositions in the ecosystem or in anywhere in the atmosphere, or I mean, biosphere consists of all these variabilities. I mean, you have, let's say, for example, carbon is 98.8% is all 12 carbon, and it's about 1.2% or 1.1% of carbon is 13 carbon. But if you look at various different kinds of things, the extent of this particular compositions vary tremendously. Like for example, the atmosphere has some value like this here. We call it as -8 because it is compared with that of an international standard. It is depleted in the 13 carbon. Whereas when you look at the C4 plants, somewhere here, C4 plants have more enriched 13 carbon than the C3 plants, which we have here. And the CAM plants will span over a whole lot of them, explaining kind of physiology that we look at, which we don't have to worry. But what I'm trying to say is you have a large variation in the composition of the stable isotopes of carbon. You have the variation in the stable isotope of oxygen, various components that could be water or the organisms, the shells, the trees, the herbaceous plants, and so on. And we have a lot of variations in the oxygen isotope in the precipitation that we get. For example, the precipitation that we get in oxygen isotope of the rainfall that we get in Bangalore is very, very different from that of what you get in Rahuri or what you get in Delhi. So if you look at the isotopic composition of your hair, I will tell you which water have you been drinking. So, I mean, it can be that accurate. That story is very interesting. Let's not worry about that now. But if I use this strategy for understanding the plant sciences, and that's where the interest is for us. And the same thing is true for nitrogen isotopes. All the 3 elements that we look at from the biological point of view have tremendous variations in the nature, and then we can pick up that as a strategy for understanding the variations in the traits that we are looking for specifically. Now, what I said was to summarize the composition of these isotopes are quite different among the organic and inorganic substances, yes, but this difference has come because of any biochemical or biophysical process that occurs that helps in the formation of these end products, bring about this kind of differences in the composition. Now, if you can understand what has happened to this particular isotopic composition, we will be able to understand what was the process and how better the process or how inferior the process was in individuals. Now, to show this, I have a small cartoon here. If I press this, I hope this works. I've never tried this in a webinar situation, but I think it should work. Now, what we have is on the left side, you have dry ice, which is carbon dioxide solid, and it sublimates and then forms carbon dioxide gas. But this is a physical process. And then we have a chemical reaction where carbon is in the form of coal. And that coal, when you oxidize, forms carbon dioxide. So you have a physical process and a chemical process. And you have 2 different types of isotopic compositions of this. You have the heavy isotope, which is a little bigger one. And then the bluish one is the smaller round, smaller circles, or the lighter isotope of carbon. Now you can see that the composition is very heavily skewed toward the lighter isotope. But what happens if you allow that to sublimate? You can see that the number of the heavy isotope depletes, it decreases when you have, or when you sublimate the physical process, or when you oxidize as a reaction that you have, which is a chemical reaction process. Whether it is a vapor pressure driven process or the reactivity that happens between some molecules by the chemical process or physical process, in all the cases, the end product is almost always depleted in the heavy isotope. And the extent of depletion of the heavy isotope will tell us how efficient the system has always been. Therefore, any process that involves either make a bond between two molecules or atoms or break the bond between two molecules will now change the composition of the isotopes. Therefore, the product will be quite different or distinct in the isotopic composition when compared to the reactant. Therefore, we can use that as a strategy to make measurements on certain specific traits that we are looking for. If I look at water use efficiency as a trait, water use efficiency is governed by two very important processes. I don't think I have that slide, but nevertheless, so I can explain that. When you look at water use efficiency, you have two very important traits that control this. On the numerator, you have the photosynthetic carbon assimilation, and in the denominator, you have transpiration. If you decrease the transpiration, the ratio, that is water use efficiency, will increase. Or if you increase the photosynthetic rate, then there is an increase in the in water use efficiency. Whether you increase water photosynthesis or you decrease the transpiration, the internal CO2 concentration decreases. I mean, this don't worry about this now for the time being. But whenever there is a reduction in the isotope in the substrate concentration, then the system will discriminate less, in the sense it does not. When it's something like when you don't have anything at all, you will eat whatever that you get. But the moment you have 100 different dishes, you will go there and choose whatever you want. That's exactly what the plants do. And therefore, there is a very, very strong inverse relationship that exists between the carbon isotope composition, which we call as the carbon isotope discrimination made by the plant, and the water use efficiency. And this has been proved in a large number of species. I mean, our own data has both perennial and annual species, and a very, very large body of literature exists, which anybody can see, anybody can read and understand this. Now, this is one thing that we have. And did anybody use this strategy for crop improvement? Has there been a challenge? This was pioneered by the Australian group. I was glad that I worked with this man, Professor Graham Farquhar, for my postdoc, and his colleague Richard Richards, who visited my lab, and even Graham Farquhar visited our lab recently. And then we started developing this kind of a strategy for crop improvement. The Australians way back in 2004 did this. They developed varieties of wheat for the dryland agriculture system of the Western Australia, which is very, very dry. And they developed precisely based on the phenotypic strategy of selecting for carbon isotope ratio. They developed better yielding varieties under stress. But what they realized, I mean, this is done using a instrument called the mass spectrometer or isotope ratio mass spectrometer. And we have that facility, and someone who wants to visit, we're welcome. And then we can also work with us to measure samples. Now, when we look at the importance of that particular variety that they developed in Australia, the yield advantage, which we have in the y-axis here, decreased when the amount of water in the soil became better. That means the yield advantage of a drought-tolerant variety was seen only when there is drought, but not when water content, water availability was high. And that's not a good idea at all, because what we look for in the breeding is the drought-tolerant variety should yield well under drought, but the yield decrease must not be significantly high when the water content in the soil increases or when the stress is removed. And that is the challenge, real challenge. And let's see how science can help us to understand this. So what we mean is to now try and understand improving the water use efficiency without decreasing the denominator or without decreasing the transpiration is the challenge. So what we wanted to do was to now look at what people have done in the literature. And then there was a lot of science that people had done in terms of improving, understanding how the evaporation occurs. You know, when water is evaporating, the H2O is evaporating, but which H2O is going isotopically is a question. Deuterium containing water or oxygen 18 containing water is heavy water, so that stays back without evaporation. And when evaporation occurs rapidly, the enrichment of the oxygen isotope in the water that stays back is always high. People have done that in the oceans, and people have shown that when you look at the oxygen isotope of the water that you have in the soil, I mean in the ocean water, you will know how much water has evaporated, and that they calculate to find out how much water it is going to rain in the monsoon and all those kind of things. This is a brilliant study that people have done in geology, and way back in 1965 they published this. This is called the Craig and Gordon model, and we extended that for a little ocean called the Leaf. I mean, you don't have to have a Pacific Ocean, and the ocean that we have in the Leaf is good enough to now tell us. What we did was to now understand the oxygen isotopic composition of the leaf water and the leaf biomass, and we proved that that has a very strong relationship with water use efficiency in carbon isotope, and the oxygen isotope relates to stomatal conductance or the mean transpiration rate of the plant. Now we have two tools which can be used, and therefore the oxygen isotope can also be used as a surrogate for measuring the differences in the root rates. We use these techniques, and we wanted to now improve the water use efficiency of crop plants. Then when we now went around and then identified the kind of species or maybe in cropland that we were interested in, then we said that is the water use efficiency a potential trait at all for drought tolerance? The answer is yes. The Australian people have shown, but our study says no, it is not good at all, because from the breeder's point of view, if you increase a particular trait, the biomass should increase or yield should increase, but it was not the case. There's only a scatter. And this was actually very, very disappointing to us about 10 years ago. And then we started analyzing this entire thing. We realized that there are many things that are responsible for this. One of the major things that happens in the field is the root. Now, if you look at the genotypes of rice or groundnut, a large number of about 300 germplasm of rice are used for this. And then we look at the low root genotypes when compared to high root genotypes. The high root genotypes had less water use efficiency than the low root genotypes. The low root genotypes are efficient because they're not accessing water, they're not losing water, therefore the denominator is changing. And therefore, whenever water use efficiency increases, it is because of a reduction in the transpiration and biomass. And Abraham Blum, who was one of the strongest proponents against the use of water use efficiency, kept saying this. But when we did that analysis to understand this, what we did was we removed the variations in leaf area, we removed the variations in the root traits. In the genotypes which have comparable roots or water or the leaf area, then the carbon isotope ratios or the water use efficiency strongly related to biomass and yield. It was not a scatter. Therefore, now if you did that kind of an analysis, our answer could also be yes, along with the Australians. Therefore, initially, the analysis that we need to do is to remove the kind of variability that brings about the other kind of differences. And that led us to a challenging program to now cross the two genotypes which had differences in these two traits, root traits and the water use efficiency, to be brought together. And we did an elaborate experiment. It was started way back in I think 2000. I think 2008 or somewhere. And then when we crossed these two genotypes, Tanu is a variety which had high water use efficiency, and a germplasm line or initial evaluation trial line 15963 was a root genotype. We developed recombinant inbred lines, and we developed the QTL map by the biparental mapping population, and we identified certain specific recombinant inbred lines which were good, and we measured the all lot of physiological traits. And the line that we had, the trait introgressed lines that we had much better photosynthesis, much better stomatal conductance. But what is interesting is it had higher water use efficiency than the either of the parents. And that is interesting. That means there is a possibility of increasing the water use efficiency even if the water used by the plant is high. And this variety was recognized, and then we released it as a upland variety for aerobic cultivation in Karnataka. And this is now, we are trying to now see if it can be grown in other places through a multi-location from the All India coordinated trials. It went up to the advanced varietal trial too, and I do not know what happened to that afterwards. But I think that's what I was saying. The yield as such has a lot of genotype and environment interaction, but at a specific location, this works very beautifully. So Daksha was nominated at the national level in 2018. I'm very happy about that. And when we look at the other traits that you see, the roots and water use efficiency, there's no doubt that they bring in a tremendous amount of improvement. And we said that there is some other thing which the plants experience is when the plants are stressed and they acquire certain traits. And these acquired tolerance have a very, very specific advantage. The genotype that express certain genes during the milder level of stress will make that plant to protect itself from the stress when the stress level becomes severe. What we did was we took this germplasm once again and we developed a strategy called the temperature induction response. One of my students, Dr. Pushpa, did this exercise very, very nicely. And then what she did was to now use this technique to now screen them. And we identified the genotypes that had differences in the recovery growth upon stress induction. And to cut the long story short, what we do is we stress the plants with a milder level of stress, and the stress is allowed to increase to a severe level. And after the severe stress comes down, we alleviate the stress and bring them to normal level of water in the soil. And then when we did that, then the recovery growth of the genotype is measured. If the plant has a propensity to respond to the milder level of stress, the recovery growth will be much, much higher. That's the premise of this strategy. And there's a lot of science that our department has done and a lot of others have done elsewhere. Dr. Madan Pal from IARI has also done a lot of work in chickpea on this particular strategy to breed for high temperature tolerance. Now, when we look at these differences in the traits, we picked up high acquired tolerance and low acquired tolerance genotypes. We compared a lot of these kind of things. You can see that the high acquired tolerance traits have a very high, the stress susceptibility indices are very low for that. And then they have much higher yields when compared to the low acquired tolerance traits. This is what I mean to say here. And we selected two genotypes which have high acquired tolerance and compared with a variety which is actually grown under irrigated conditions like BPT 5204, which is a Sona Masuri type. And you can see that the yield levels are much higher under stress and the reduction is very high here. So that means to say that you have certain traits that can be used. And we did a metabolome, a non-targeted metabolomic analysis. And this paper of Pushpa is under review in experimental botany. And then we adapted a non-targeted metabolomic approach and we identified specific metabolites that were responsible for bringing in the acquired tolerance. What we did was to now find out the genetics of this. So this particular set of germplasm was already genotyped by using SSR markers. And then we use the same data for the analysis. Now, if you see here in chromosome 8, there was one place where two markers associated with the recovery growth rate were present within 1 centimargan apart, which was 1 centimargan apart, which was very, very close in genetic terms. But molecular biology will say that there could be something like 1 MB of genome inside that part. But nevertheless, what we did was we identified what could be the genes that are present in it. There are about something like 70 genes or something. But what is interesting to me was the genes that were responsible for two very important processes, glutamate decarboxylase and raffinose synthase. Both the genes were associated with the singular pathway of glutamine processes to the raffinose and the stachyose production, and a glutamate which is decarboxylated to form the GABA and then the ABA responsive kind of things. So one mechanism or one particular region in the genome is responsible for bringing about the osmotic stress tolerance, and that's an acquired tolerance. And if we can use these things in the breeding processes, I think we can improve the yield more comprehensively under stress. So a little more science we did. We found out what are all the other genes that were there. There was this gene associated with the oxidative stress or the ROS scavenging mechanisms of the reduced glutathione and the kind of thing that we know very, very nicely. And the ROS, the reactive oxygen species, scavenged from there were also associated with the acquired tolerance traits. Now, to put all of them together, what we did was to use this particular genotype called AC39020, which had high roots and high acquired tolerance, and a particular high water use efficiency germplasm called 8016348. And we developed a very specific multi-parent backcross strategy, which one of my PhD students, Dr. Pratibha, did this work. And she crossed them, developed the F1s, and these F1s were crossed between themselves to develop backcross, to develop the double cross progeny. And the double cross progeny were selected based on the marker that we had identified before. And those were selected and then backcrossed to IR64 three times. And the BC3 F1s were allowed to self, and she developed about 10,000 BC3 F2 lines. And from there, we selected 260 of them. Again, to cut the very long story short, we picked up some 20 genotypes that had the trait introgression. Now, the trait introgression line is the gray one, and the black one is the IR64. You can see that the relative water content of them are similar under well-watered condition, but the things come very, very evident when they are stressed. This was, I think, the stress was to an extent of about 55 to 60% of the field capacity. And then you can see that the trait introgression line, which had all the three mechanisms, that is the cellular level tolerance or acquired tolerance, roots, and water efficiency, maintained much better water relations, and they maintained much better leaf area. They maintained definitely better spikelet fertility under stress. That led to higher yields. And we did because we had a backcross progeny developed, the background genome recovery was done using 120 SSR markers. And the background of this particular genotype that Pratibha developed has about 98% of the genome of IR64. You can see that the grains of IR64 and the trait introgression lines, they look almost the same. So that means we have improved IR64 essentially. And the yield of this particular variety is about 30, maybe it's twice more than the productivity of IR64 under water limited conditions. And these are the lines that will be useful for the development for cultivation under the water limiting aerobic condition, which saves water. And these are some of those data once again to show that there are these lines are doing much better than the IR64 at even much severe levels of stress. Now this brings us to a situation where we say drought research is almost entirely phenotype centric. So we need to be very clear in terms of understanding the phenotype, and we need to have a mechanism of developing a method to measure these traits. And I think Dr. Vishwanathan tomorrow perhaps will also be talking about a fantastic facility that they have in IARI, which is called as the Nanaji Deshmukh Phenomics platform, which I'm sure most of you are aware. And what we developed is a very similar kind of a thing, but the differences are that we have things that are grown in open air here. If this video works, I'll be glad. This is a small video that we developed from here. What we do is, this is a mini lysimeter phenomics facility at UIS Bangalore. And this is indigenously developed. And then each of these containers that you look at here are sitting on a load cell balance. There is no need of moving these plants. And when the plants are evaporating or transpiring, the weight of the container decreases. Now there is a software controlled solenoid valve, and that dispenses exactly the same amount of water, so that the water level in the container can be maintained at all times in a day. So that permits us to find out how much of water is lost in the morning, how much of water is lost in the afternoon, night, and that is interesting. Nocturnal transpiration is gaining a lot of importance these days. Can you see this? Just about 1.5 minutes of a video that we developed. Now these are all the precision mechanisms, which is very similar to what our facility at IARI is. And this is a field facility where they have certain cameras which will go there and capture it. They have such facility at ICRISAT. And these are all very intensely worked out mechanisms. But we wanted to now develop an improvised platform where we keep the plants under naturally varying conditions. And then this is what I said. Each of these containers is sitting on a load cell balance. And then there's a rainout shelter which is mechanically moved by power, and that will protect the system from the rain. And this is a very indigenously developed system. That is where it is unique and definitely cost effective. All these motorized semi-automated mechanisms will help dispense water into this container at whatever the requirement is. So therefore, depending on the requirement of water, you can have very specific levels of water stress maintained in it. So you have a live display of the data right there at the system, and then there is a mechanism of calibrating the balances at all the time whenever we want. And there's something called the Internet of Things, or the kind of Wi-Fi connections where you have wireless communications in it. And this is a camera that we look for measuring the growth. And this is what I said, there's a very specific way of maintaining stress cycles in the plants. So we can quantify, presently we are doing a screening of about 150 genotypes of rice, and we have done that. This is my professor, my boss, my guru. We've done that in many species so far, in both cereals and in certain legumes. And this facility helped us to now phenotype the germplasm and the trait in progress lines, and that led to the discovery of development of this variety called Daksha. Oh yeah, that's the end of that cinema. Now, to summarize what I've been saying, there's tremendous progress has been made globally and even in India to understand the physiological basis of drought adaptation. And we have now developed and we have access in the country for phenotyping under both natural and under controlled conditions for analyzing and capturing the genetic diversity that you have in these traits. And these genetic resources, I mean, mapping populations or breeding lines or different germplasm or any of those kind of things, or mutants especially, are now available in the country, can be done. And I'm sure there are a lot of geneticists who have tremendous knowledge and expertise now in the country for the genomics-based crop improvement approaches. And I'm very sure, like in the first slide, I said by 2050, we are going to have a lot of difficulty of feeding 1.5 billion people. With the kind of progress that is being made in the country, I'm very confident that we are not going to die of hunger. That's for sure. And I should thank the funding that we received from the Government of India, from ICR, from DBT and DST, and even from our Karnataka state government. And very recently, we had a larger program on the Indo-Swiss collaboration in biotechnology for genomics of one of the millet species called finger millet. And thanks to all my young colleagues who have been working very hard. Thanks to all of them, and thank you so much for this opportunity. I'll be very happy if there are some questions. Thank you.
Thank you, sir. Thanks for your nice presentation. We have some questions in chat box.
Yeah.
I would like to ask you some questions
Sure.
from participants. Balaguru from Tamil Nadu Agriculture University. Uh, is it possible through water stress we have improved fruit quality?
Yeah. I mean, that is exactly what happens if you look at the fruits that you get, let's say grapes or anything in summer, is always tastier or sweeter when compared to one that grows in well-watered conditions. That is because the concentration of the sugars will be higher in water-limited conditions. That is, yes. But then, are we improving that is the question. Like, for example, if you look at the sugar content in a fruit which is grown in rainy season or in summer, if the fruit has the same sugar content per fruit or the sugar concentration per fruit, that is the important question. And whether the stress can improve the quality, is that what the question that is? Is a question that can water stress improve the quality? Hello?
Prashant.
Prashant? Prashant, is the question, what is the question? Is stress improving the water? Is the quality of the fruit? Is that the question?
Hello, sir. The question is it possible through water stress we have improved food quality?
Yeah. I mean, water stress is, let's say, remove a little bit of water and give a mild stress. I'm sure the quality will increase. But then, if the quantity of the fruit that you produce will decrease, and I do not know if you will, the consumer will pay more money for a sweeter apple than a less sweet apple. We don't have a mechanism of that at all. Therefore, yield is still an important issue. Yield should not decrease. Quality may increase in terms of sweetness. The quality is basically many other kind of things. Is there proteins, is there amino acids, or are the vitamins there, and are they digestible in the human being? There are a lot of questions in quality, and I'm not very sure what happens to the fruit quality under water if there is any stress. It should decrease, I think.
Okay. Thank you, sir. One more question asked by Mr. Balaguru. In root crops, how we can develop water stress resistance varieties?
In a what crop?
Root crops.
Yeah, sure. I mean, if the root is an important thing, look for the highest root fellow. Number one, this is, I mean, you can follow the same thing that we just discussed. I mean, even for the roots to grow more, you need carbohydrates to be supplied there, and that has to increase. And that can increase only when the photosynthesis increases, isn't it? So we can increase the drought tolerance in root crops also by the same strategy that we just now discussed.
Okay. Thank you, sir. Next, Professor Sanjay Bade has one query. He asked, which factor to determine the WUE and how to manage to WUE in agrochemical aspects?
What do you mean agrochemical aspect?
It's not agrochemical, it's agronomical aspects.
Agronomical. Sure. I mean, again, the same thing that I said. You know, there is a very small difference between what I, as a physiologist, define water use efficiency and what the agronomist defines it. When I say it is the amount of carbon assimilated per unit amount of water transpired by the plant, the agronomist will look at what is the yield for the amount of water that is supplied in irrigation. Both are exactly the same. In agronomic perspective, a water use efficient genotype would definitely use less water. Now, what we had done was to now give the variety that we developed by trait introgression to the agronomist, and they grew them in the field and then they irrigated it in different levels. For example, one of the agronomy experiment that was done was very, very nice. They grew in the puddled condition where there was standing water, and then they gave it certain specific amounts of water, which was, the amount of water that was very measured and given. And then there was a drip irrigation mechanism. And then there was something which was left to rain-fed situations. If it rains, it grows, otherwise it will die. So the rain-fed rice definitely did not do well at all. But what was very, very interesting to me was that the varieties that we developed, or the one that had trait introgression, from the agronomic perspective, definitely performed better. The variety was only second to a hybrid that he used in the field. So hybrids were doing well, both under well-watered or puddled or in the the upland situations or the aerobic situation. But our variety definitely did well. That was the beautiful part of it. So agronomically, there is a chance, there's a possibility that we can measure this, and this will definitely do. The water use efficiency that we talk about in physiological terms can be now studied under the field levels for agronomic conversions of it. It works. I hope I answered that question.
Okay. Thank you, sir. The next, Mr. Chander has a question. How to improve productivity in off-season in mango?
I don't know. I mean, I worked in rice. I think I'm not very sure about how to increase the yield under off season. Why do you want that? I mean, they say that you should eat seasonal fruit. There are a lot of fruits available around the year. I mean, there's no doubt that you should increase the mango, like how you can grow something like banana and all that all the time. But then I don't know. I'm not very sure. I don't think I'm qualified to answer that question in how to do that in mangoes.
I think Dr. Vishwanathan Chinnaswamy has answered the earlier question also. That partial root zone drying is used to induce mild drought in grapes for increasing antioxidant and anthocyanins for wine and color parameters also. So
Okay.
for fruit crops.
All right. Thanks. That's very nice, Vishwa.
Sir, I have one question. If
Yes, sir.
Can we breed for improved carbon isotope mechanism through normal breeding, or transgenic is the only option for that?
No. Whatever we have done is not all transgenic. It is definitely possible to do it by breeding. I mean, this particular stable isotope that I told you is only a phenotyping strategy. We are only measuring the differences in water use efficiency, transpiration, and productivity by using stable isotope as a rapid tool. That's all. But we do that by breeding. Definitely, yes. We don't need any transgenic at all for this.
Okay, thank you. Dr. Deshmukh? Prashant, any other questions?
Thank you, sir. Sir, nowadays everybody is working on transcription factors. What is your opinion about stress levels and transcription factors?
Oh, I mean that's there's a lot of use in that. You know, when we look at this, Prashant, when you we know that the expression of any trait is at the genetic level, and the gene has to express. And the gene expression, as I said, that there are two types of traits: constitutive traits, and the one that are responsive traits, or what we call the acquired traits. Now, these acquired traits or responsive traits only can respond to a stress, and that means they don't express during the time when there is no stress, but the moment when there is stress, there is a big increase in the activity of such genes. This is possible only when transcription factors are doing the job to now express certain genes very specifically. Therefore, transcription factors have a long way to go. I mean, that's a very beautiful what you call as mechanism that one can improve to make the plants to sustain better under stress. If you can understand the transcription activity, there's no doubt about the importance of that. You know, and then there are a large number of these transcription factors already discovered: drought specific, heat specific, and then combined ones. And then what happens under cold and abiotic, and even when there is a pathogen infection, there are a large number of them. There is a very recent discovery about a transcription factor which helps in providing broad spectrum resistance against blast disease in rice, against a pathogen fungus called Pyricularia oryzae or Pyricularia, whatever that species is. That fungus is causing a big important disease. And this particular broad spectrum resistance comes to the plant from a very specific transcription factor. So there's a lot to discover, and there's a lot of scope for transcription activation in plants for developing resistance to stress for sure.
Thank you, sir. Bhaveshri asks one question: is there any change in nutrient accumulation in the seeds of genotypes with a different root system architecture?
Yeah. See, basically the roots are the ones that are absorbing both nutrition and water. And if the nutrients are present in the soil, the better architecture of the root will definitely help accumulation of better nutrients in the grains. That we have seen, yes, and that is an important thing to do, and that's a good question. Yes, it happens. If the root system architecture and the root activity is better, absorption of nutrient and its accumulation in the grains from the human perspective will also be important. Yes, there's no doubt about it.
Hello, sir. I have a question.
Yes, sir.
Dr. Sheshshayee. Today, in the today's scenario, much is talked about the combination of stress, particularly combination of abiotic stresses.
Yes.
So the developed variety with better water use efficiency, is it also tested under heat stress?
Yes.
Or, yeah.
Yes, sir. Yes, see the
combination of the
correct. See, there are two questions. One is when we look at water use efficiency as a mechanism, that mechanism will now say how much carbon was fixed for a unit amount of water that was transpired by the plant. But if we are looking at what happens to the mechanisms of photosynthesis or mechanisms of transpiration under high temperature stress or under heat stress, then we will also be looking at the mechanisms of whether or not the traits that are responsible for water use efficiency are also related to the high temperature mechanisms. There are a lot of reviews that are coming about now. The under high temperature, the process of photosynthesis or the mechanisms of carbon diffusion, they will definitely get affected. Though water use efficiency cannot be called as a trait for high temperature stress, but we can look at transpiration as something which will make the plants cooler, which will have to now, for example, thermal imaging that people are doing now, infrared guns are there which we can measure the temperature of the canopy. The canopy must remain cooler for the metabolism to take place properly. So a trait that is associated with maintaining the cooler canopy will be very important for high temperature stress or heat stress. But the heat stress may have a very different influence on the metabolism itself. So from that point of view, there can be a very small connection between water use efficiency as a drought tolerant mechanism and for heat. But then there are very specific mechanisms for heat as well.
thank you. Okay.
Thank you, sir. Thank you, sir, for nice presentation and answers to the participants' questions. Many questions are there.
Yeah.
However, we can try to solve these questions uh, afterwards with the help of expert. Now, I would like to request Dr. Lokhande sir to the, take vote of thanks for the expert.
Very good morning to everyone. I profusely thank Honorable Vice Chancellor Dr. K.P. Vishwanatha sir, Dean and Director of Instructions Dr. A.L. Farande sir, Director of Research Dr. Yasar Gadak sir, Co-Convener and Principal Investigator CST CAST CSAW MPK Veravari Dr. S.D. Gorantiwar sir, for approving the organization of this webinar. Respected Associate Dean, PGI, Dr. P.N. Rasal, sir, Co-Principal Investigator, Dr. M.D. Shinde, sir, Organizing Secretary and Head Department of Biochemistry, Dr. Aryam Naik, sir, Joint Organizing Secretary, Dr. P.L. Kulwal, sir, and all the participants from national and international organizations. It is my privilege to propose the vote of thanks on this webinar. I, on the behalf of this university and organizing committee, I extend deep sense of gratitude to Dr. M.S. Sheshshayee, Professor, Department of Crop Physiology, University of Agricultural Sciences, Bangalore, for having accepted our invitation and for giving in-depth knowledge how to improve the crop productivity and grain quality in changing climate. Thank you once again, sir. I extend my sincere thanks to Dr. Prashant Kale, Dr. Nandalal Deshmukh, and Engineer Mohsin Tamboli for their untired efforts to make the session successful. I thank all the participants, faculty and students from national and international organizations for actively participation in webinar. With the permission of organizing secretary and co-convener, shall we close the session, sir?
Thank you very much for this opportunity. I have always enjoyed discussing. Except if I were to, unlike a present situation where we could go there and meet each other and discuss for a long periods of time. This is only short, but it's okay. But that way, the only difference is that I have to have my own coffee. Otherwise, we would have enjoyed the coffee from your place. Thank you so much for this opportunity, and I enjoyed it.
Thank you, Dr. Sheshshayee.
Thank you, sir. Thank you, sir.
For a wonderful presentation.
Thank you, sir.
Thank you, sir. Dr. Prashant.
Okay, thank you, sir.